So biomolecules, or as you may prefer to call them, biological molecules, molecules are these substances that are produced by cells and organisms. There are four main types: proteins, carbohydrates, lipids and nucleic acids, and most of these are made up of small monomer molecules joining together to form a polymer. So for example here you have an amino acid, which is the monomer unit of a protein. On the right hand side there's a carboxyl group, and then on the left hand side there's an amino group, which you could probably guess because amino acid, amino group. You also have a variable region. This is different between all the 20 naturally occurring amino acids, which is what makes them varying. And then you just have a hydrogen group stuck on the top. So this structure is consistent between all amino acids, it's just the variable bit that varies. And then you have a central carbon. Because this is attached to all different groups, it's called an alpha carbon. And just a little bit more information, despite them all having the same basic structure, because of the variable group, all the amino acids have different physical and chemical reactivities. And then here's just a little diagram of all our essential amino acids. These are essential to consume because we can't generate them ourselves within the body. And then these guys are our non-essential amino acids, which we don't need to take in. In this bit, it's just a table of all the amino acids and the 3 and 1 letter codes. This is often used in, I think it's called the codon table. It's what bases pair up to form which amino acids. You can see it in a table. And then this little pie chart here is actually showing what proportion of each sort of biomolecule and molecules that humans are made up of. And actually we're made up of quite a bit of protein, so humans are made up of 16% protein. And there are about 6 main types of proteins. So you've got catalysts, which are enzymes. These basically catalyze chemical reactions. You've got signaling proteins, which are your hormones and receptors. They control signals and coordinate activity. You've also got transport proteins, such as hemoglobin. This is used to transport oxygen or carbon dioxide or carbon monoxide or dissolved substances through the blood. You've got structural, such as muscle and tissue. These enable structure and mobility. You've got defensive proteins, such as your antibodies, which defend against foreign bodies. And then you've got storage proteins, such as, I can never say this, ammublin. However you pronounce that, which is egg whites. These are biological reserves of metal ions and amino acids, which are the building blocks of life. If you did biology at A level or maybe even at GCSE level, you'll know that your proteins are made up of amino acids. So a protein is a polymer of the monomer amino acids, and these amino acids join together via condensation between an OH group of the carboxyl group and a H group of the amino group. This forms a peptide bond, and so a peptide bond formation by dehydration is the removal of the OH and the H group to form the bond. So that means you have a OH group and a H just sort of out in the wild, and these combine to form a water molecule, which is your H2O was seen there. The one that was just in the previous slide section bit was a dipeptide. That di meaning 2 and peptide meaning peptide. There's also oligopeptides, which consist of 3 to 10 amino acids, and polypeptides, which can be chains of 300 to 1,000 amino acids, and these are what are proteins. And there are 4 basic different types of structure that you need to know about. Well, levels of structure. First is your primary, which is the number and sequence of amino acids in a polypeptide chain. At this point, it's not classed as being 3D or 3-dimensional. You've got your secondary, which is a localized conformation of the chain. This occurs through folding due to the into beta-pleated sheets and alpha helixes due to the different charges on amino acids groups and their interactions. You've got tertiary, which is further folding of a polypeptide, but this time it is into a specific three-dimensional structure. These can be compact and globular, or long and spindly, which are fibrous. You've also got your quaternary or fourth level. This is when multiple tertiary structure polypeptide chains join together. This isn't through any form of covalent bonding. It occurs due to, again, the electrostatic interaction between the different groups of the amino acids. And here's just a little example of hemoglobin, which is a quaternary structure protein. So as I mentioned earlier, proteins can be catalysts, which are used in reactions, and it's important to remember that reactions occur in steps. They don't just sort of all happen at once. And these biological catalysts, which are proteins, are called enzymes. So enzymes increase the rate of chemical reactions without being used up, so they can be reused. And it's important to remember they only affect the rate of the reaction, not the amount of product formed. They do this through altering the transition state. So as you can see here, the red line is showing a reaction without an enzyme. More activation energy is required to catalyse the reaction, whereas in the blue line with an enzyme, there's less activation energy required to catalyse the reaction, so it occurs faster. But there's some difficulty with enzymes. They are very specific, so one enzyme catalyses one reaction due to its highly specific active site, and they're very sensitive to things like temperature and pH because, again, they're proteins, so they can easily denature. However, inorganic catalysts can work over a range of different temperatures and pHs. Here's just a little diagrammatic explanation of what I mentioned earlier about the enzyme being reformed. So you've got your enzyme and your substrate binding together to form an enzyme-substrate complex. The catalytic step then occurs with this molecule and it reforms the enzyme and forms the product. And then here's a little diagrammatic explanation about the high specificity of enzymes. So you can see here, its active site is that weird U shape, and only the substrate that's a weird U shape can fit in it, as long as it has the correct orientation and is aligned correctly with it.